So the scheduled time would be starting 5:30, but already one is video presentation and another one presenter is already here. So I would like to start to that final session of that oxidative stress. Would you please starting that video or that Dr. Yoshiaki Harakawa? Yeah, please. Thank you.
Hello, I am Yoshiaki Harakawa from Division of Antioxidant Research, Gifu University. First, I'd like to thank you for the opportunity to give a presentation today. I would like to start my presentation. The title is Potential of an Antioxidant Supplement for Amyotrophic Lateral Sclerosis. Amyotrophic lateral sclerosis, or ALS, is an intractable disease in which the muscle of the limbs, throat, and tongue, and the muscles necessary for breathing gradually become thin and lose strength. It causes motor impairment, dysphagia, and respiratory failure. Instead of affecting muscle itself, ALS affects the motor neurons that control muscle movement and exercise. In ALS, the brain's command to move the limbs are no longer transmitted, causing the muscles to weaken. Normally, all other bodily sensations, vision, hearing, and internal organ functions are usually preserved. ALS is commonly seen in men as male patients are 1.3 to 1.5 times more common than female patients. It usually develops after middle age. In Japan, 2.2 people per 100,000 people contract the disease each year, and there are about 10,000 patients as of 2020. The cause of ALS are not yet fully understood. Approximately 90% of all cases of ALS are considered to be sporadic ALS, for which the cause is unknown. It has been reported that misfolded SOD1 with an abnormally folded structure is involved in the pathogenesis of sporadic ALS. On the other hand, about 5 to 10% of all ALS cases are known as familial ALS, which is hereditary from patient to child. It has been reported that one of the main causes of this is due to point mutation in the SOD1 gene. Oxidative stress damage caused by SOD1 abnormalities is thought to be deeply involved in the pathogenesis of ALS. In addition, it has been reported that the level of multiple oxidative stress markers, such as 808DG, 4-HNE, and ascorbate free radical, are elevated in cerebrospinal fluid samples from ALS patients. Therefore, it is thought that oxidative stress is involved in the progression and deterioration of ALS. And ALS and other neurological diseases are linked to mitochondrial dysfunction and oxidative stress. Familial ALS gene mutations such as SOD1 have been found to induce mitochondrial dysfunction and increase the production of ROS, suggesting a role for oxidative stress in the disease progression. Alzheimer's disease, the most common form of dementia, has been shown to cause mitochondrial dysfunction and oxidative stress, due to the accumulation of beta amyloid in the brain. Other diseases such as Parkinson's and Huntington's disease also show mitochondrial dysfunction and oxidative stress. Although the pathological features and clinical symptoms of each disease differ, they can be said to share a common theme on their onset. Oxidative stress were introduced earlier. Here's a brief recap. The source of oxidative stress is reactive oxygen species and free radicals, and excess of ROS can lead to cellular inflammation. An antioxidant counteracts the free radicals and ROS, maintain the balance. The balance of such ROS and free radicals is disrupted when they increase excessively or when antioxidant activity is weakened, and this code is called a state of oxidative stress. One option for improving oxidative stress is to take antioxidant. If ALS is an oxidative stress disease, could antioxidant be a treatment option for ALS? The Edaravone introduced here is a drug developed as a treatment for acute cerebral infarction. In cerebral infarction, ischemia and reperfusion cause the generation of free radicals such as hydroxyl radicals, which are known to cause cell membrane damage and brain dysfunction by oxidizing unsaturated fatty acids in cell membrane lipids. Edaravone, as a radical scavenger, eliminates free radicals such as hydroxyl radical and suppresses lipid peroxidation. This suppresses oxidative damage to brain cells. For Alzheimer's disease, it has been found to significantly improve motor and cognitive impairment, reduce the accumulation of amyloid beta, p-tau, and alleviate neuronal loss, oxidative stress, and neuroinflammation. Although this drug was originally for cerebral infarction, it was proven in multiple human clinical trials that it suppressed the decrease in the revised ALS functional rating scale compared to the placebo group by inhibiting lipid peroxidation reaction with its radical scavenger ability. As a result, in June 2015, Edaravone was approved as the world's first therapeutic drug to show efficacy in inhibiting ALS progression. Twendee X, as I introduced earlier, is an antioxidant supplement developed by Professor Inufusa of our division. It has been proven to have high oxidant activity and safety with 8 types of active ingredients, including vitamins and amino acids. Here we will discuss the possibility of applying this Twendee X to ALS treatment. The antioxidant property of Twendee X were examined in vitro. It is known that when cells are exposed to hydrogen peroxide, reactive oxygen species are produced. Hydrogen peroxide treatment of cultured cells resulted in a 69% increase in mitochondrial ROS and 68% increase in cellular ROS. In addition, mitochondrial SOD and intracellular SOD decreased by 32% and 31% respectively. This was accompanied by an increase in the GSSG-GSH ratio. The addition of Twendee X reduced hydrogen peroxide-induced ROS and increased the antioxidant levels in the cell and mitochondria. This suggests that Twendee X has a protective effect on mitochondria due to its antioxidant property. In order to investigate the effect of adding Twendee X, we also conducted a metabolome analysis using the same cell line. Cells that were not treated with Twendee X were used as a control. Cells were stimulated with Twendee X for 1 hour. The red areas in the heat map indicate area where expression is increased. This region is mainly involved in mitochondrial energy production, and it was found that Twendee X stimulates energy production. The data shown here is from the literature by Dr. Koji Abe. In Alzheimer's disease model mice, Twendee X administration reduces the accumulation of amyloid beta in the brain compared to the control. In addition, significant decrease in multiple inflammatory and oxidative stress markers such as IL-1 beta, TNF-alpha, OHNE, and 8-OHDG were confirmed. Furthermore, in an all-diabetic mice model, the administration of Twendee X suppressed the decline in autophagy function more than in the control group. In addition, Twendee X has been proven to prevent the decrease in neurogenesis in the brain of aged mice caused by aging. With the above effect, Twendee X has been reported by Dr. Koji Fukui of Shibaura Institute of Technology to significantly improve motor and cognitive impairment in aged mice models. So far, we have been examining Twendee X in vitro and in animal models. In order to examine its effectiveness in preventing dementia in humans, a multi-center randomized placebo-controlled double-blind clinical trials were conducted. This study targeted patients with mild cognitive impairment in Japan who took either Twendee X or placebo for 6 months, and changes in dementia symptoms from the baseline over 6 months are examined. As a result, the subject taking the supplement showed significant improvement in the dementia scores, the mini mental state examination, and the Hasegawa dementia scale compared to the placebo group. This study has revealed the clinical efficacy of Twendee X on the cognitive function of MCI subjects, and it has been suggested that powerful antioxidant treatment may be useful for preventing MCI, which is prone to developing into Alzheimer's disease. Based on these results, the Evidence Creation Committee of the Japan Society for Dementia Prevention has certified Twendee X as effective in preventing dementia. The certificate is shown in this slide. From these data, let's consider the potential of Twendee X for the treatment of ALS. ALS is characterized by mitochondrial dysfunction and oxidative stress, which are also common to other neurodegenerative diseases such as Alzheimer's disease. Twendee X protects and activates the mitochondria through its strong antioxidant property, reduces amyloid beta accumulation, inflammatory and oxidative stress markers in the brain, and maintains autophagy and neurogenesis. It has been proven to be effective in preventing dementia in humans. Therefore, Twendee X is thought to be a promising option for the treatment of ALS. Here's a conclusion. ALS is a severe neurological and oxidative stress disease. There have been several reports of the potential of antioxidants to treat ALS. Edaravone is a pharmaceutical radical scavenger. It was originally approved as a treatment for acute cerebral infarction, but is already being used in clinical practice to suppress oxidative stress and neuroinflammation in ALS. Twendee X has been proven to have antioxidant properties and corresponding mitochondrial protective function, and it has also been shown to be effective in the treatment of Alzheimer's disease, another neurodegenerative disorder. Twendee X is a dietary supplement. Therefore, it can be taken without prescription or the concern of side effects, which is a huge advantage. Supplementation of antioxidants can be considered as a treatment option for ALS. That is all. Thank you for listening.